Good afternoon, everyone. I am Dr. Satyabrata, MDDM clinical pharmacologist, and today I am going to discuss on a new topic, that is the pharmacological effects of nitric oxide. So, coming to introduction, nitric oxide, first described in 1979 as a potent relaxant of peripheral vascular smooth muscle. It is used by the body as a signaling molecule, and it serves different functions depending on body system, that is neurotransmitter, vasodilator, bactericide. It is an environmental pollutant, and it is the first gas known to act as a biological messenger. Then coming to the structure and nature of nitric oxide. Nitric oxide is a diatomic free radical consisting of one atom of nitrogen and one atom of oxygen. It is lipid soluble and very small for easy passage between cell membranes. It is short lived, usually degraded or reacted within a few seconds. Then coming to synthesis of nitric oxide. Nitric oxide written as NO or simply NO. NO dot or simply NO. A highly reactive signaling molecule made by any of three close related nitric oxide synthase isoenzymes, each encoded by a separate gene. And these isoforms generate nitric oxide NO from the amino acid L-arginine in an O2 and NADPH dependent reaction. This reaction involves enzyme bound cofactors including heme, tetrahydrobiopterin, and flavin adenine dinucleotide, that is FAD. So, in the case of NNOS, that is neuronal nitric oxide synthase, and ENOS, endothelial nitric oxide synthase, NO synthesis is triggered by agents and processes that increase cytosolic calcium concentrations. NNOS and ENOS are activated by calcium. Another is INOS, that is indissoluble nitric oxide synthase, is not regulated by calcium. Then coming to the properties of the three isoforms of nitric oxide synthase. So, NOS1, NOS2, NOS3. So, other names, NOS1 is NNOS, that is neuronal NOS, NOS2 is INOS, that is indissoluble NOS, NOS3 is ENOS, that is endothelial NOS. So, NOS1 found in neurons, skeletal muscles, NOS2 in macrophages, smooth muscle cells, NOS3 found in endothelial cells, neurons. Then NOS1 expression, it is constitutive. NOS1 is constitutive, NOS2 is transcriptional induction, NOS3 is constitutive. Then calcium regulation, depending on calcium regulation, NOS1 yes depends on calcium, and NOS2 no, it doesn't depend on calcium. NOS3 yes, it depends on calcium. Then coming to signaling mechanism, nitric oxide mediates its effects by covalent modification of proteins. There are three major targets of nitric oxide. One is metalloproteins. NO interacts with metals, especially iron in heme. The major target of NO nitric oxide is soluble guanylyl cyclase (SGC), a heme containing enzyme that generates cyclic guanosine monophosphate, cyclic GMP (CGMP) from guanosine triphosphate, that is GTP. NO nitric oxide binds to the heme in SGC, soluble guanylyl cyclase, resulting in enzyme activation and elevation in intracellular CGMP, cyclic GMP guanosine monophosphate levels, and CGMP, cyclic guanosine monophosphate, activates protein kinase G, PKG, which phosphorylates specific proteins. In blood vessels, NO nitric oxide dependent elevations in CGMP, cyclic GMP, guanosine monophosphate, and PKG, protein kinase G activity result in the phosphorylation of proteins, leading to reduced cytosolic calcium levels and subsequently reduced contraction of vascular smooth muscle. Then interaction of NO nitric oxide with other metalloproteins mediates some of the cytotoxic effects of NO nitric oxide, for example by activated macrophages. For example, nitric oxide inhibits metalloproteins involved in cellular respiration like aconitase and cytochrome oxidase. Then thiols. Thiols, nitric oxide reacts with thiols to form nitrosothiol. In proteins, the thiol moiety is found in the amino acid cysteine. This post-translational modification, termed S-nitrosylation or S-nitrosation, requires either metals or O2 to catalyze the formation of nitrosothiol adduct. S-nitrosylation can alter the function stability. Major targets are HRAS, activated by S-nitrosylation, and metabolic enzyme glyceraldehyde 3-phosphate dehydrogenase inhibited by S-nitrosylation. Glutathione can be S-nitrosylated to generate S-nitrosoglutathione. Then tyrosine nitration. NO, nitric oxide, undergoes both oxidative and reductive reactions, resulting in a variety of oxides of nitrogen that can nitrosylate thiols and nitrate tyrosines. Nitric oxide NO reacts with superoxide to form peroxynitrite ONO that leads to DNA damage, nitration of tyrosine, and oxidation of cysteine to disulfides or sulfur oxides. Numerous proteins are susceptible to peroxynitrite catalyzed tyrosine nitration. Glutathione can protect against tissue damage by scavenging peroxynitrite. This is the image showing the nitric oxide mechanism and how it is released and how it works, and the cysteine is nitrosylation, tyrosine nitration, etc., and how arginine gives rise to NO. And you can go through the book to learn more about this image. And then coming to nitric oxide in disease. The vascular effects of nitric oxide. Nitric oxide has significant effect on vascular smooth muscle tone and blood pressure. Endothelium dependent vasodilators such as acetylcholine and bradykinin, by increasing intracellular calcium levels in endothelial cells, lead to the synthesis of NO, nitric oxide. So nitric oxide diffuses to vascular smooth muscle, leading to vasorelaxation. This is another image that shows the mechanism of nitric oxide and also how it is synthesized from arginine, involvement of ENOS, and how it is calcium dependent, and how acetylcholine bradykinin leads to NO release, and how NO acts on soluble guanine cyclase to produce cyclic GMP, and how phosphodiesterase degrades cyclic GMP to guanosine monophosphate, and cyclic GMP leads to activate the PKG, and that is inhibit the contraction, so in the vascular smooth muscle cell. So there is two cells, one is endothelial cell, another is vascular smooth muscle cell, and how calcium is released from endoplasmic reticulum, and so this is all about the nitric oxide. You can go through the image from the books to learn more about this image and this topic. The nitric oxide also has antithrombotic effects. Both endothelial cells and platelets contain ENOs, endothelial nitric oxide synthase, which acts via an NO cyclic GMP pathway to inhibit platelet activation, an initiator of thrombus formation. Thus, in diseases associated with endothelial dysfunction, decrease in nitric oxide generation leads to thrombosis. And nitric oxide has an additional inhibitory effect on blood coagulation by enhancing fibrinolysis via an effect on plasminogen. Nitric oxide also protects against atherogenesis. A major anti-atherogenic mechanism of NO, nitric oxide, involves the inhibition of proliferation and migration of vascular smooth muscle cells. In animal models, Myointimal proliferation following angioplasty can be blocked by nitric oxide donors by NOS nitric synthase gene transfer and by NO inhalation, nitric oxide inhalation. Nitric oxide reduces endothelial adhesion of monocytes and leukocytes, lead to atheromatous plaques due to inhibitory effect of NO nitric oxide on the expression of adhesion molecules. In addition, nitric oxide may act as an antioxidant, blocking the oxidation of LDL, low density lipoprotein, and thus preventing or reducing the formation of foam cells in the vascular wall. Plaque formation is also affected by NO nitric oxide dependent reduction in endothelial cell permeability to lipoproteins. Then in septic shock, infection causes sepsis. Endotoxin components from bacterial wall induce synthesis of INOS in macrophages, inducible nitric oxide synthase in macrophages, neutrophils, etc. NO nitric oxide causes hypotension, shock, death. Knockout mice lacking a functional INOS inducible nitric oxide synthase gene are more resistant to endotoxin than wild type mice. Then infection and inflammation. Nitric oxide NO has both beneficial and detrimental roles in host immune response and in inflammation. NO nitric oxide stimulates the synthesis of inflammatory prostaglandins by activating COX-2 and cyclooxygenase 2. Nitric oxide NO contributes to the erythema, vascular permeability, and subsequent edema associated with acute inflammation. Nitric oxide production by dietary L-arginine supplementation exacerbates arthritis, whereas protection is seen with INOS, inducible and nitric oxide synthase inhibitors. The central nervous system. Nitric oxide has an important role in the central nervous system as a neurotransmitter. Nitric oxide is not stored in the synaptic vesicles but is synthesized on demand and immediately diffuses to neighboring cells. Nitric oxide synthesis induced at postsynaptic sites in neurons. Nitric oxide may function as a retrograde messenger and diffuse to the presynaptic terminal, helps in learning and memory. NMDA receptor activation, N-methyl diaspartate receptor activation, and excessive NO nitric oxide synthesis is linked to excitotoxic neuronal death in several neurologic diseases including stroke and Parkinson's disease. Therapy with NOS, nitric oxide synthase inhibitors, may reduce neuronal damage in these conditions. The peripheral nervous system. NANC, non-adrenergic, non-cholinergic neurons are widely distributed in peripheral tissues, especially the GIT, gastrointestinal tract, and reproductive tracts. Nitric oxide is a mediator of certain NANC, non-adrenergic, non-cholinergic actions, and some NANC neurons appear to release nitric oxide. Penile erection is due to release of nitric oxide from NANC neurons. Nitric oxide promotes relaxation of smooth muscle in the corpora cavernosa. Sildenafil, tadalafil, vardenafil used in treating erectile dysfunction by releasing nitric oxide. Respiratory disorders. Nitric oxide administered by inhalation to newborns with hypoxic respiratory failure associated with pulmonary hypertension. Nitric oxide inhalation dilates pulmonary vessels, resulting in decreased pulmonary vascular resistance and reduced pulmonary artery pressure. Inhaled nitric oxide also improves oxygenation by reducing mismatch of ventilation and perfusion in the lung. Inhalation of nitric oxide results in dilation of pulmonary vessels in areas of the lung with better ventilation. Nitric oxide inhalation does not typically exert pronounced effects on the systemic circulation due to the enrichment of PDE, phosphodiesterase E5, in pulmonary vascular beds. PDE5 inhibitors such as sildenafil and tadalafil induce vasodilation and marked reductions in pulmonary hypertension. Then coming to pharmacologic manipulation of nitric oxide. Inhibitors of nitric oxide synthesis. The primary strategy to reduce nitric oxide generation in cells is to use NOS inhibitors, nitric oxide synthase inhibitors. The majority of these inhibitors are arginine analogs binding to the NOS nitric oxide synthase arginine binding site. In inflammatory disorders and sepsis, inhibition of the INOS, individual NOS nitric oxide synthase isoform, is potentially beneficial. NNOS, neuronal NOS nitric oxide synthase specific inhibitors, useful in treatment of neurodegenerative conditions. Administration of non-selective NOS nitric oxide synthase inhibitors leads to concurrent inhibition of ENOS endothelial nitric oxide synthase, resulting in vasoconstriction and potential ischemic damage. Some inhibitors of nitric oxide synthesis or action: LNMMA, LNAM, 7-nitroindazole, PBS2, hemoglobin. So these are written the mechanism and comment of these inhibitors. So you can go through the book to learn more about this. Then nitric oxide donors. Nitric oxide donors which release nitric oxide or related nitric oxide species are used to elicit smooth muscle relaxation. Different classes of nitric oxide donors are organic nitrates, that is nitroglycerin, which dilates veins and coronary arteries, is metabolized by NO nitric oxide by mitochondrial aldehyde reductase. Venous dilation decreases cardiac preload. Combining with coronary artery dilation accounts for the antianginal effects of nitroglycerin. Other organic nitrates such as isosorbide dinitrate are metabolized to an NO releasing species. Organic nitrates. Organic nitrates such as inhalant amyl nitrite also require metabolic activation to elicit vasodilation. Nitrates are arterial vasodilators. Amyl nitrite is abused for euphoric effects and if combined with phosphodiesterase inhibitors can cause lethal hypotension. Amyl nitrite has been largely replaced by nitrates such as nitroglycerin, which are more easily administered. And sodium nitroprusside. Sodium nitroprusside, which dilates arterioles and venues, used for rapid pressure reduction in response to light as well as chemical or enzymatic mechanisms in cell membranes. Sodium nitroprusside breaks down to generate 5 cyanide molecules and a single NO nitric oxide. Then NO gas inhalation. Inhalation of NO nitric oxide results in reduced pulmonary artery pressure and improved perfusion of ventilated areas of the lung. Inhaled NO nitric oxide is used for pulmonary hypertension, acute hypoxemia, and cardiopulmonary resuscitation. Inhaled NO nitric oxide is stored as a compressed gas mixture with nitrogen. Nitric oxide can react with O2 to form nitrogen dioxide, a pulmonary irritant that can cause deterioration of lung function. Additionally, nitric oxide can induce the formation of methemoglobin, a form of hemoglobin containing Fe3+ rather than Fe2+, which does not bind O2. Therefore, O2, that is oxygen. Therefore, nitrogen dioxide and methemoglobin levels are monitored during inhaled NO nitric oxide treatment. Then alternate strategies. Another mechanism to potentiate the actions of NO nitric oxide is to inhibit the phosphodiesterase enzymes that degrade cGMP, cyclic guanosine monophosphate. Inhibitors of type 5 phosphodiesterase such as sildenafil result in prolongation of the duration of NO nitric oxide induced cGMP, cyclic guanosine monophosphate elevations in a variety of tissues. So that's all about effects of nitric oxide. So thank you for watching my videos, and you can go through the books and articles to learn more about this topic. So thank you very much. Jai Jagannath.